Hello, this is Dr. Divya Pradhan, and today we are going to talk about organic molecules. Okay, so what are organic molecules? Organic molecules always include two elements, carbon and hydrogen. Carbon and hydrogen form these long chains and they're called hydrocarbons. Not necessarily true, because organic molecules can be made up of other elements and we call them CHNOPs, C-H-N-O-P-S, carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. These may or may not be present in all the organic molecules, but carbon and hydrogen, yes, they will prevail in all organic molecules. Okay, so what allows this variety of organic molecules to be formed is because carbon has four valence electrons, and these valence electrons can share electrons with either other carbon or with any other atom. So why does it do that? It will share electrons, remember, to satisfy the octet rule. We've talked about the octet rule, right? So in this way, a variety of organic molecules are formed. What is the second most important characteristic of organic molecules? The functional groups. So all organic molecules have certain functional groups. Look at this, hydroxyl or carbonyl. So first of all, what's a functional group? These are specific combinations of bonded atoms that always react in the same way. So an acid will always react in the same way. A amino will always react in the same way. A sulfhydryl will always react the same way, or a phosphate group will always react the same way. Okay. The third important characteristic, even though organic molecules are large, have a very, very high molecular mass, they are very simple to understand because they're all made up of monomers, repeating subunits. So a good example would be a pearl necklace. So instead of studying the entire pearl necklace, just study individual pearls. So a pearl necklace will be a polymer. Poly means many, mer molecule. Just study a monomer, single molecule. All right. So here are the important biomolecules, and this is an important slide. So what are the polymers? What are the monomers? For carbohydrates, the polymer is called a polysaccharide. Monomer is called a monosaccharide. Lipid, the polymer is fat, monomer is glycerol and fatty acid. For protein, excuse me, polymer is a polypeptide and a monomer is amino acid. And for nucleic acids, the polymer is DNA or RNA and a monomer is a nucleotide. All organic molecules, they have a common reaction. If you want to synthesize, then you'll go through a dehydration reaction, removal of a molecule of water. So monomer, each are going to give up OH and H. So one is going to give up OH, one is going to give up H. You'll have water and you'll form a, what is it? A dimer or a polymer, right? For a polymer to break down, you go through hydrolysis. Add water, so one will accept hydrogen, one will accept OH, and you will have two monomers, all right? So the first one that we are studying is, of course, a monosaccharide. So monosaccharides are sugars. Mono means one, saccharine means sugar. These are with 3 or 7 carbon atoms. This, of course, is a glucose. And you can see there are 6 carbon atoms, 1, 2, 3, 4, 5, and 6. You can depict it either way, or this way. This is the one that's most preferred. Disaccharide, here you can see glucose and glucose join by which process? Dehydration process to form maltose, which is a di, means 2. Saccharine means sugar. Polysaccharides, of course, are long chains of monosaccharides, and here they are for 2 functions, energy storage or structural. Okay, here is the energy storage, the first one of a polysaccharide, which is starch. It's a polymer of glucose, and here with just some branching, otherwise it's kind of straight chain, okay? So here is glycogen. Glycogen is another polysaccharide. This is animals store energy in the form of glucose in the form of glycogen. Plants store glucose in the form of starch, okay? This is branched polymer of glucose molecule. These are very highly branched. There are two types of structural polysaccharides, cellulose and chitin. Cellulose forms the cell wall of plants and maintain or give the support, because obviously plants do not have a backbone like us. And chitin is typically seen in insects or also in fungi, forming the cell wall. Again, the way the glucose, again the glucose is joined, but the way the glucose is joined is different for chitin or cellulose or starch or glycogen. All right. Lipids. Lipids contain more energy per gram than any other biological molecules, but one common characteristic, hydrophobic. That means they do not dissolve in water. They are water hating. There are three types: fats and oils, phospholipids, steroids. Fats are of animal origin. They are solid at room temperature and either store energy or insulate, and of course provide protective cushion. Oils are usually plant origin and they are liquid at room temperature. Here is a typical fat molecule. So for a fat molecule you have glycerol, 3 carbons, 1, 2, 3, and to each of this carbon molecule you attach a long chain of fatty acid. Acid will have what? COOH. So here when you attach this, remember what process we are going to go through? Dehydration process. That means there's a loss of water and you will have a fat molecule. The way I remember it is as an E. Backbone is glycerol and here are the 3 chains of fatty acid. Alright, there are 3 types of fatty acids. They're saturated, unsaturated and trans. Let's see. Saturated fatty acids, as you can see, have no double bond, alright, just single bond. Is unsaturated fatty acid has double bond or a triple bond. So this is an unsaturated fatty acid. And finally you have the phospholipid. Phospholipid, if you remember the glycerol molecule, the 3 carbon chain, this is attached to a this is attached to a fatty acid, not this. This is attached to a fatty acid chain, and this is attached to a phosphate molecule. This is going to be very important when we'll talk about cell membranes. Steroids. Steroids have a backbone of 4 fused carbon rings. Very important when we talk about hormones, especially the sex hormones or the stress hormones, testosterone and estrogen. And of course there are more adrenal cortex produces, or the adrenal gland produces more hormones, and so those are the of steroid in nature. And then there is a cholesterol, which is also important for the cell membrane formation. Proteins. Proteins are polymers of amino acids, and here the amino acid is there's a carbon, and the carbon is attached to an amino group, to an acid group, to a hydrogen group, and of course we have the R, which is the alkyl group, which determines the uniqueness of the amino acid. Alright, so here as you can see there are 4 common amino acids, each have a different R, and because of which they of course have different chemical properties, and these are 4 are very important biologically. Now proteins perform different functions, and what are they? They are structural, enzymatic, hormones, actin and myosin, transport, antibodies for protection, and channels. We'll talk about this to some extent in Bio 103, but of course details are found of course in higher classes. Nucleic acids. 2 types of nucleic acid, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Alright, so if you see the structure of DNA, remember DNA and RNA are polymers. What's the monomer? The monomer is a nucleotide made up of a nitrogen containing base, a sugar and a phosphate. So phosphate and sugar is constant. The nitrogen containing base is going to be different, as you can see in the next slide. So here the DNA, the sugar is deoxyribose, and here it is ribose. You don't have to learn the structural characteristic, but this difference you will have to know. The bases, what are those nitrogen bases? Remember I talked about the nitrogen bases right here. Well, the nitrogen bases are adenine, guanine, thymine, and cytosine. In RNA, adenine, guanine, cytosine remain the same. Thymine is replaced by what? Uracil. There are strands are double strands for DNA, single strand for RNA. Sometimes there are, of course, this a few differences, a few times that the RNA becomes double-stranded, but you don't need to worry about that. And helix. What are we talking about helix? Helix is this. They're coiled like this. And there's helix for DNA. There's no helix for RNA. All right, here's the structure of a DNA. You can see the double helix. You can see the backbone, and here are the nitrogen bases. And the nitrogen bases form hydrogen bonds with the complementary base pairing. You don't need to know about it right now. We'll talk about that in the other slides. Here's again repeated. Here is the structure of DNA, where cytosine binds or bonds to guanine, the two different chains. Remember I told you there are two chains, complementary chains, and here's the bonding form, almost like a ladder. All right, and here the last of the nucleic acid is the ATP, and the most important is adenosine triphosphate. As you can see, 3 phosphates. And what happens is that when you need energy, this last phosphate molecule breaks off, and you will have ADP, adenosine diphosphate, and as a result, you will have liberate energy. All right, and I think, yes, with this we come to the end of our presentation. I thank you for your time and attention. And if there are questions or concerns, please stop by my office.